Students, hope you all are fine. As a part of your biochemistry core biochemistry paper in proteins, till now we have come across 20 naturally occurring amino acids, the structural features of these amino acids, and special features and properties exemplified by each of these 20 naturally occurring amino acids, and of course many more things. Let us move on to the next part of our discussion. Our today's topic of discussion is peptide bond and its special features. After the end of this lecture, after listening to this entire talk, I think that all of you would be in a state so as to come up with answers to very few important questions about peptide bond. That what do you mean by a peptide bond? What is the type of biochemical reaction which generates a peptide bond? Then what are the special biochemical features of a peptide bond? What are the special geometrical features or structural features of a peptide bond, and so on. So to begin with, let us take an introductory look over peptide bond. We know that proteins are actually the polymers of many amino acid residues which are linked together by a linkage called a peptide bond or an amide linkage. How is such a bond formed, and what is a peptide bond in reality? That is in biochemistry. Peptide bond is formed when two amino acids interact with each other in course of a biochemical reaction. The carboxyl group during this interaction, the carboxyl group of one amino acid attached to its central carbon interacts with the basic amino group of the second amino acid that is again attached to the central carbon of the second amino acid. In course of the process, the basic amino group of the second amino acid acts as a nucleophile, and it is capable of making an attack over the acid carboxyl group of the first amino acid. So the NH2, that is the basic amino group, makes a nucleophilic attack on the acid carboxyl group of the second amino acid to displace or to release hydroxyl ion in the form of OH- from the carboxyl group. Thus, the two participating chemical moieties which participate to form a peptide bond are the acid carboxyl group and the basic amine group. The bond is actually a condensation reaction between carboxyl group and basic amine group to generate a partial double, that is partial covalent bond, so-called amide linkage. The COOH group is a leaving group and donates its OH- ions, while the NH2 group donates H or hydrogen, and thus the elements of water in the form of H and OH- donated by NH2 and COOH are released to generate or to link the two moieties of the two amino acid residues together through a CONH peptide linkage, which is also called amide linkage. Hence, it is a condensation reaction in which, to summarize, OH- acts as a nucleophile, and OH- makes a nucleophilic attack over the carboxyl group of the first amino acid, and in turn the basic amino group of the second amino acid acts as a nucleophile, makes a nucleophilic attack over the acid carboxyl group of the first amino acid and releases OH-, and thus OH- of the COOH group acts as a leaving group and finally generates a peptide linkage. Likewise, in a tripeptide containing three amino acid residues, the amino acids are linked by two peptides. An oligopeptide would usually consist of about 8 or nearly 8 amino acid residues linked together by peptides, and hundreds of amino acid residues get linked together gradually to form what is called a protein or a polypeptide. Now let us take a look over few special features. So now it's quite clear that the peptide bond is the CONH amide bond which links to amino acid residues. Let us take a look over come across few important special features or biochemical features and structural or geometrical features of the peptide bond. The peptide bond, scientists like biochemists from the very beginning have been conducting model building experiments, have been conducting X-ray crystallographic diffraction experiments to study the biochemistry and the geometry of peptide bond. So such experiments were initially conducted by two prominent biochemists, Pauling and Corey, and the results, along with results, Pauling and Corey could conclude few important things about peptide bond. That the peptide bond present in CONH is, or that is the CN bond, is shorter than the bond is shorter than a primary amine. Thus, and this indicates of a kind of resonance, that is partial double bond nature or polar covalent nature of a peptide bond. I'll be coming into it again. Now the second, number one, the first important feature is that peptide bond is actually having is rigid and coplanar and has partial double bond character. Another important feature of peptide bond is that all the atoms of the peptide group lie in a single plane. Now the question arises that how many atoms make up a peptide bond? Altogether, the bond is CONH, made up of 4 atoms, C, O, nitrogen and hydrogen. And besides, apart from these, there are 2 central anomeric carbons or asymmetric carbons of 2 amino acid residues, and altogether these that is summate to produce 6 atoms which are present in a peptide group. Hence, we can say that 6 atoms of the peptide group lie in a single plane, or each peptide bond consists of 6 atoms with the carbonyl oxygen and the hydrogen of the nitrogen lying trans to each other in opposite directions or in opposite sides directions of the planes, or being trans to each other. This is another important feature of peptide bond. Hence, and one more feature is, now let us talk a bit about the resonance character of a peptide bond. In a peptide bond, it's shorter than the bond present in CN, that is primary amine, and this was primary indicative of resonance. Further experiments or investigations came up with a very interesting observation that in peptide bond there occurs a share of lone pair of electrons between the carbonyl oxygen and the hydrogen of the nitrogen. The carbonyl oxygen of the carbonyl group acquires in course a partial negative charge, and the hydrogen of the nitrogen in course of this sharing of lone pair of electrons acquires a partial positive charge. The 2 oppositely charged poles together set up an electric dipole, thereby making the peptide bond coplanar, or making it acquire partial double bond character. Hence, the CN peptide bonds, rotation about CN bonds, peptide bonds have a double bond character, and rotation is not permitted around the peptide bonds, and the presence and this feature of peptide bond limits the conformation of the bond peptide, that is polypeptides or proteins in which such bonds are present. So now let us summarize a few essential features. Number 1, the peptide bond is rigid and coplanar. Number 2, it shows resonance. Number 3, it is shorter than the syn bond present in simple amine. Number 4, all the 6 atoms of the peptide group lie in a single plane. Number 5, it has an electric dipole, shows a double bond character, and hence rotation is not permitted about CN bonds. Rather, rotation is permitted about NCC alpha and CC alpha, 2 more bonds. We'll study about these bonds in our future classes or in our next class. So for today we can that is what we have, the gist of what we have studied is that we studied about peptide bond, how is it generated, how are the elements of water released, the condensation reaction which generates the peptide bond, few special biochemical and geometrical or structural features of peptide bond. Next day, we'll study about the NC alpha and CC alpha, that is bond angles of peptide bond, rotation around those, and about the Ramachandran plot. As of now, for today, as a part of your work, I would suggest you all to go through the biochemistry of the peptide bond, the geometry of the peptide bond from the book Leninger Principles of Biochemistry. I would suggest that's a part of the bibliography which I would like to mention over here. So Leninger should be the book which you should go through very thoroughly, and you should study and stay prepared for your next classes. See you all in the next classes. Till then, goodbye.